Welcome everyone. In this chapter, we are going to study carbohydrate. We will learn the definition, classification, chemical properties, important monosaccharide and disaccharide, polysaccharide, like starch and glycogen, and also the common qualitative tests for carbohydrate. Before starting, let us see what we will cover in this chapter. We will learn the definition of carbohydrate and their classification with example. Then we will study chemical properties like oxidation, reduction, and oxygen formation. After that, we will understand monosaccharide like glucose, fructose, and galactose. Then we will learn disaccharide like maltose, lactose, and sucrose. Next, we will study polysaccharide like starch and glycogen. Finally, we will learn important quality tests and the biological role of carbohydrate. Now let us understand the definition of carbohydrate. A carbohydrate are polyhydroxy alcohol which contain an aldehyde group or a ketone group and also their derivatives. Carbohydrate contain carbon, hydrogen, oxygen. Usually hydrogen and oxygen are present in the ratio of 2:1, similar to water, so they were called hydrate of carbon. The general formula is written as CH2ON or CmH2ON. Carbohydrates are very important because they provide energy, help is in storage of energy, and they also play a role in lubricant, immunity, and cell-to-cell communication. Energy in the body. For example, sugars and starch provide energy quickly. In our body, glucose can be stored as glycogen in the body, liver, and muscles. Carbohydrates also help in protein and fat metabolism. Some carbohydrates like cellulose provide structure to plant. So carbohydrates have both energy and structural importance. This slide shows the general structure of carbohydrates. The carbohydrates contain many hydroxyl groups, which means there are polyhydroxyl. They also contain one carbonyl group, which can be either an aldehyde group or a ketone group. Because of this group, carbohydrates can take part in different chemical reactions. Now let us see the classification of carbohydrates. The carbohydrates are classified mainly into four groups: monosaccharide, disaccharide, alkosaccharide, and polysaccharide. Monosaccharides are the simplest unit. Disaccharides are made up of two monosaccharides. Alkosaccharides are made up of 3 to 10 monosaccharides. Polysaccharides are made up more than 10 monosaccharide units. Monosaccharides are the simplest unit of carbohydrate. They cannot be further broken down into the smaller carbohydrate. Examples include glucose, fructose, and galactose. These are very important in the body because they are the basic energy molecule and also the building block for bigger carbohydrates. Disaccharides are formed when two monosaccharides join together by a bond called a glycosidic bond. This happens by a condensation reaction where one water molecule is removed. Important disaccharides are maltose, lactose, and sucrose. Oligosaccharides contain 3 to 10 monosaccharide units. An example is maltodextrose. These are less common as major energy source but They are important in cell recognition and some biological function. Polysaccharide contain more than 10 monosaccharide and can be very large. They are mainly used for storage of energy and structural function. Examples are starch, glycogen, and cellulose. Carbohydrates can also be classified as aldoses and ketoses. If the carbonyl group is an aldehyde group, it is called an aldose. For example, glucose is aldose. If the carbonyl group is a ketone group, it is called a ketose. For example, fructose is a ketose. Carbohydrates can be classified as reducing sugars and non-reducing sugars. Reducing sugars have a free aldehyde group or a free ketone group is solution, so they can reduce reagent like Benedict solution. Examples are glucose, lactose, and maltose. Non-reducing sugar do not show this property. A common example is sucrose. Polysaccharide can be classified as homopolysaccharide and heteropolysaccharide. Homopolysaccharide are made up of only one type of monosaccharide. Examples are starch and cellulose. Heteropolysaccharide are made of more than one type of monosaccharide. Examples include heparin and hyaluronic acid. Now let us study the chemical properties of carbohydrates. Carbohydrates show chemical reaction mainly because of the carbonyl group and hydroxyl group. Common reactions include oxidation and reduction to alcohol and formation of oxygen. These chemical properties are also the basic for mainly laboratory tests. First, let us understand oxidation. Reducing sugar like glucose can be oxidized to form carboxylic acid. For example, in phonetic test, glucose is oxidized to gluconic acid. That is why glucose is called a reducing sugar. This oxidation reaction is used in lab tests to detect reducing sugar. Next, carbohydrate can undergo reduction to alcohol. The carbonyl group in the open chain form can be reduced to an alcohol by reagent like sodium borohydride or catalytic hydrogenation. The product formed are called aldehydes. This, uh, this is an important chemical property of monosaccharide. Next, carbohydrates can form oxygen. Oxygen are formed when reducing sugar react with excess phenyl hydrogen. This produces crystalline derivatives. For example, glucose form glucosogen. Oxygen formation is also used in laboratory identification of sugars. Now let us study the structure of monosaccharide. The monosaccharide have the general formula H2O(N). In their structure, one carbon has a carbonyl group. If the carbonyl group is at the end of the carbon chain, the sugar is an aldose. If the carbonyl group is in the middle of the chain, the sugar is a ketose. Monosaccharide can join together to form bigger carbohydrate like disaccharide and polysaccharide. Now we study glucose. The glucose is most important monosaccharide in the human body. It is the main energy source for cells. Glucose is broken down during the glycolysis to produce energy. The body excess glucose is stored as glycogen in liver and muscles. In plant, glucose units join together and form cellulose, which provide strength to the plant cell wall. Now we study of fructose. Fructose is fructose sugar. It is found naturally in fruit and honey. Fructose can join with other monosaccharides to form oligosaccharide. A very important example is sucrose. Sucrose contains one glucose and one fructose joined by a glycostatic bond. Now we study galactose. Galactose is an important monosaccharide found mainly in milk. It joined with glucose to form galactose, which is called milk sugar. Lactose stores energy in its bond. To digest lactose, the body needs a special enzyme. If this enzyme is not present, lactose intolerance and occurs. Now we move to disaccharide. Disaccharide are sucrose made up to monosaccharide joined by a glycosidic bond. The three most important disaccharides for our body are maltose, lactose, and sucrose. Maltose is made up of two glucose molecules and is found in germination seed. Lactose is made up of glucose and galactose and is found in milk. Sucrose is made up of glucose and fructose and is called by table sugar. Now we study polysaccharide. Polysaccharides are also called glycan. They contain more than 10 monosaccharide units and can be very long when they are hydrolyzed. They produce many monosaccharide molecules. Polysaccharides differ based on the type of monosaccharide, chain length, bond type, and branching. Polysaccharides namely have two important functions: energy storage and structural support. Polysaccharide can be homopolysaccharide or heteropolysaccharide. Homopolysaccharide contains the same monosaccharide repeated, like starch, glycogen, cellulose, and pectin. Heteropolysaccharide contain different monosaccharides like hyaluronic acid and chondroitin. These are important in connective tissue and lubrication. Now let us understand glycogen. Glycogen is called animal starch. It is the storage form of glucose in animals. It is found namely in liver and muscles. Glycogen has a structure similar to starch but has more branching. Because of branching, glycogen can be broken down quickly to release glucose when the body needs energy. Now we study cellulose. Cellulose is a structural polysaccharide found in plant cell walls. It provides trunk to plant. Unlike starch and glycogen, cellulose form a linear polymer and give strong fibrous structure. Humans cannot digest cellulose because we do not have the enzyme to break it bind, so it acts as dietary fiber. Now we study starch component, amylose and amylopectin. Amylose is linear chain compound of starch and deep blue color with iodine. Amylopectin is a branched chain compound of starch and a purple color with iodine. So iodine test can help identify starch and also give information about starch compound. Qualitative test for carbohydrate. These tests are important in lab and also for exams. Different tests can identify carbohydrate and also help in distinguish between monosaccharide, disaccharide and the type of sugars. First is Shelvanov test. This test helps to differentiate between maltose and ketose sugars. In this test, ketose shows like fructose from a jelly red complex after reaction. So fructose gives a pathway result faster than glucose. This test is useful to identify ketose sugar. Next is Barford test. This test is similar to Benedict test, but it's done in an acidic medium. Monosaccharide react faster than disaccharide in acidic medium. Barford reagent contains copper acetate in legible acetic acid. So this test helps to differentiate monosaccharide from disaccharide. Next is spelling test. The test is also based on the reduction property of sugars, similarly to Benedict test. Spelling test reagent has two parts. Spelling A contains copper sulfate and spelling B contains sodium potassium tartrate. Spelling test the pathway result for maltose sugar due to aldehyde group. Ketose sugar can also give positive result because they can convert to aldose form under alkaline condition. Next is Mole's test. The Mole's test is a general test for carbohydrate. In this test, carbohydrates are dehydrated by strong acid to form purpural derivatives. These derivatives react with alpha naphthol and produce a purple color ring at the junction of two layers. A purple ring indicates a pathway test for carbohydrate. This is oxygen formation test. All reducing sugar from oxygen when reacted with excess phenyl hydrogen at boiling temperature. Different sugar from different shaped crystals. For example, glucose, fructose, galactose and mannose form needle shaped oxygen. Maltose form sunflower-like crystals, so this test helps in identification of sugar. Next is Benedict test. The Benedict test detects reducing sugar. When reducing sugars are heated with Benedict test reagent and alkaline medium, they reduce cupric ions to cuprous ions, forming a red precipitate to have copper oxide. Any sugar with free aldehyde or ketose group can produce Benedict reagent. This test is semi-quantitative because the color of precipitate gives rough estimate of sugar concentration. In Benedict test, the color change gives an approximate idea of the amount of reducing sugar present. Green color indicated low amount, yellow show moderate amount, orange show higher amount, brick red indicate very high amount. So Benedict test is useful to detect and actually estimate reducing sugar in urine or a sample. To summarize, carbohydrates are important molecules that provide energy and also help in storage and structure. They study the definition, classification, and chemical properties of carbohydrates. We learn important monosaccharide and disaccharide and also polysaccharide like starch and glycogen. Finally, to cover important qualitative tests like molecular test, genetic test, link test, barcode test, Sullivan of test, oxygen formation and iodine test. This completed chapter 2.